Welcome to FNL Technical Service video series. Today we have Dr. Christine Fennessey from Viral Evolution Core in our AIDS and Cancer Virus program. Dr. Fennessey, welcome and thank you for talking to us today about unique services your lab provides. Please tell us a little bit about services that you offer.
Well, first of all, our core offers single genome amplification services, or SGA for short, which basically means we can sequence SIV, HIV, or SHIV genomes on an individual genome level. So we can take a variety of sample types, tissue, cells, plasma, culture supernatant, and then we isolate the viral DNA and RNA, and we dilute these samples down so that there's only a single genome to act as template for a PCR reaction. So we're able to then amplify a single gene or a whole region or sometimes the whole genome from this single template, and then we perform sequencing. And then we perform a rigorous QC process before submission to our collaborators. This kind of sequencing is incredibly useful if you have samples that have very low viral loads in them, and if you're looking for specific mutations or sequences in a mixed population that you wouldn't necessarily be able to see if you performed bulk sequencing of your sample. This is also useful for if you're looking for specific paired mutations within a genome that might be also impossible to see with bulk sequencing.
Thank you for that. So what makes your services unique?
Our core is unique in that we try and communicate with our collaborators really closely. Before even starting the project, we have a conversation to make sure that we choose the best possible protocol to answer their research question. And once we've finished all the sequencing and QC of the project, we also like to be as heavily involved in the analysis as the collaborators want us or need us to be. We're always available to help make sense of the data we generate. For example, we often prepare amino acid translations from our data or make phylogenetic trees, highlighter plots. We provide a lot of resources to turn our raw sequencing data into something that's accessible and applicable to answering the research question. In addition, we're also happy to help produce publication quality figures and methods. We've been doing this for a while now, so we definitely have experience in how to best represent this kind of data in a publication.
Thank you. That definitely sounds unique enough. And one final question for you is, what type of research or researchers do you normally support?
We've provided our services to a broad variety of collaborators, usually from academic and government labs. And our core is also frequently used by investigators within our program, as well as by external collaborators. But we've applied this SGA technique to a huge range of research projects, from vaccines to viral evolution to transmission. We're happy to help anyone who might think this service might add value to their project.
Thank you. Dr. Fennessey, thank you so much for talking to us today.
My pleasure.
And if you're interested, in learning more about these services, please feel free to contact us. Thank you.
You're welcome. Thanks.